Pleasure to introduce Dyann Wirth, Harvard T.H. Chan School of Public Health, to talk to us about the next step, which is, of course, expanding the malaria toolkit. Thank you.
Thank you very much, Gina and Chris, for the invitation to speak at this important and memorable honoring of Alan McGill and his vision. Chris and Regina asked me to talk about kind of this idea of using basic science to expand the toolbox. And so that's what I'm going to focus on, and I'm going to focus on this using a vision that I think Alan had in terms of how science can be applied. So in terms of progress in the last decade in basic science, we have made amazing progress, both in our understanding of the basic science of the organisms involved, the parasite, the mosquito, and the human host, and in the technologies that we can apply to address this problem. So there is enormous hope. And I will highlight a few of these. I can't do justice to everything, but this meeting is filled with new discoveries by many of our up-and-coming young researchers, and I encourage all of you to attend as many of those sessions as possible. Now, as Alan looked at this, and Bruno, in fact, used this a related slide, Alan really looked at elimination and eradication through an evolutionary lens. That is, the idea that as parasites interact with their two hosts, it's sort of an arms race. Everybody wants to survive, and everyone works to their advantage to live the best life, to be transmitted in the case of the parasite, to survive in the case of the mosquito, and to survive in the case of the human host. And so there's this constant pressure and evolution. And we're going to talk about that. We're going to use that lens to look at basic science discoveries. So Alan really wanted us to incorporate this thinking in everything we did in elimination and eradication. And I think that's very important for us to consider. Now, Bruno's already mentioned the two kind of prominent evolution or selective processes that we know about, and I'm going to talk in some detail about those and new approaches to how to overcome those. But then I'm going to move toward the middle and end of the talk to talk about evolutionary or selective pressures where they certainly exist, but we don't understand their origin. So as Bruno mentioned, and this is again a slide from Alan's deck that he used for his presidential address, that in fact emerging resistance is one of the biggest challenges and certainly one of the results of evolution. The two resistances are drug resistance. In this case, I'm going to talk about artemisinin resistance and insecticide resistance or pyrethroid resistance. I'm first going to turn to the insects. We know that not only this is one of the obstacles to eradication. We can see it coming. There's resistance, but in addition, the measures that we currently have to deal with insects, for the most part, only deal with the indoor biting mosquito. And so, in fact, anything else escapes control. And so one basic science strategy, which I think supersedes the use of insecticides or chemical approaches, is to actually genetically engineer the parasite. And over the last 3 or 4 years, this has become possible in a very facile way. And this is the process of genetic engineering in which we can see gene drive or genes or particular selectable genes moving through populations. And so I'm going to show you just one example of that. Here is the idea where you can actually modify the genome of the mosquito either by introducing something which could kill the parasite. So you would, and then replace the mosquito population containing this, with the mosquito population containing this gene, or alternatively, you could introduce sterility and suppress the population of mosquitoes. Both of these have been tried, and there's enormous progress. This field is moving quickly. There are several sessions here. And so the concept is that by engineering sort of a beginning mosquito, that this trait, every time the mosquito mates, all of the progeny will carry this trait, and eventually, over a relatively short period of time, the population is replaced. This has enormous possibilities, but of course, there are issues of ethical issues, technical issues. Is it going to work? But this gives us hope that this is a completely different way of approaching insects by engineering the insects themselves. Okay, now to drug resistance. We all know that drug resistance is a problem. It has been in the history of malaria and in the history of infectious diseases. Technological advances made the possibility of identifying the mechanism by which parasites became resistant to artemisinin going fast forward. From the discovery of slow-clearing parasites to the identification of one of the genes involved took only a few years because of high-tech approaches of cell culture and high-throughput sequencing, coupled with excellent field work. In the work of Arie et al. And Mioto et al., they were able to identify the gene that's associated with resistance, and in fact, and now there are surveillance programs to identify this and make sure wherever possible to slow its spread. However, resistance is an evolutionary true fact. No matter what we do, the parasite's going to develop resistance. So in fact, the basic science approach is in fact to generate much more information, much more opportunity to develop new approaches to drug discovery. And there's been an international effort over the last now decade to screen millions of compounds for the ability to kill not only the blood stages of the parasite, but liver stages and transmission stages. And this, in fact, was one of Alan's sort of key visions, and you're going to hear a talk later in this session about one of those potential developments. But in fact, this phenotypic screening, this way of identifying compounds that kill the parasite, identifies lots of compounds. And this is from the work of the MD-TIP consortium led by Elizabeth Winsler at UCSD, in which a group of investigators have been screening, have been taking the compounds that were positive to kill the parasites and identifying the targets of those compounds to have a much better picture. This harnesses the technology of cell culture, high-throughput technologies, and high-throughput genomic sequencing, coupled with, CRISPR or other genetic technology and crystallography to really begin to accelerate the discovery of new targets and potentially new drugs. I think Elizabeth's vision for this is that, in fact, the entire genome of the parasite will be annotated both for druggable targets and for many of the new resistance mechanisms which are appearing under this set of studies. Of course, this works, almost all of this work is focused on Plasmodium falciparum, but in fact, one of the biggest challenges to malaria eradication from a biological sense is the Plasmodium vivax hypnozoite. That parasite exists in the liver and remains dormant, and it's experimentally, until very recently, has been inaccessible for drug discovery or drug development or basic biology. But recent work in model systems, in humanized mouse systems by Stefan Kappa's group, and what I'm going to show you in the next slides in using bioengineering and livers on a chip by Sagita Bhatia's group, it's now possible for both of these groups have identified hypnozoites or small forms in vitro culture. And this really allows them to now begin to investigate potential drug targets, but also to investigate the biology and understand this critical obstacle to eradication. Okay, So now I'm going to step back and look at evolution a little bit more broadly. And one of the things that many of us who've been involved in using genome sequencing, it is really the enabling of genome sequencing to sequence now thousands of genomes of the parasite, that we're really beginning to understand the evolution of the parasite at its most fundamental level, at the level of DNA sequence. And I just show you a slide from one of our first papers in this area, in which we looked at all of the predicted genes of the parasite and asked how diverse are those genes. And most of them have very little diversity, but then there's this very long tail that shows very high diversity. That is, parasites from around the world or even from in a region are quite different from one another. And I've plotted here the approximate location of the various vaccine targets that were in development at this time in 2007. So one of the things we predicted, and Chris Plow has been involved in this work for one of the antigens, is that in fact this data told us that pre-existing in the population of parasites in the world were parasites that were divergent, and perhaps if you vaccinated with a single allele, you would only see a protection against that allele or closely related alleles. And so, in fact, we were able to directly test this hypothesis in a collaboration with the large-scale RTSS vaccine. This work is published. I won't go into it in any great detail, except to say that we looked at every parasite escapee, every parasite that occurred either in the vaccinated or the control arm of the vaccination trial with the Broad Institute using a technology developed for high throughput and multiplex sequencing. So we examined every parasite in those individuals. And together with Peter Gilbert's group, using analytical technology developed for HIV vaccines, we tested for a sieve effect, asking whether parasites that carried the same allele as the vaccine, the RTSS vaccine, were selected or removed from the population as shown in this kind of cartoon diagram, so that the red parasites, in fact, are eliminated from the population. And the data from that trial clearly showed a significant sieve effect for the homologous strain. And so again, this idea that diversity in the parasite population has enormous implications for vaccine implementation. So there are many strategies to get around this. You could use a strategy that's used in the area of flu vaccines, combining different allelic types, or in many bacterial vaccines, or an alternate strategy, which I really think wanted to highlight here, is to actually concentrate on some aspect of the parasite biology. I'm going to show you work from Julian Rayner's group in which he concentrated on molecules that are involved in the invasion of the merozoite into the red blood cell. He was able to identify through a sort of medium throughput technology a molecule that was invariant, the RH5 molecule, and when he raised antibody to this molecule, he could show that it cross-protected against many strains with from many different geographic origins, and this gene, in fact, is almost monomorphic, or it has limited diversity. So this is an alternate strategy. Pick a vaccine candidate that doesn't show diversity in natural populations. Okay, so the final evolutionary point I want to make is kind of related to what both Pedro and Bruno showed, this concept that we are really bending the curve. We are changing the kind of frequency of malaria parasites in the world. And that has some consequences. I mean, you can see it in these very nice heat maps from the Atlas project. You are seeing reduction in the intensity or the prevalence of malaria. But in fact, that is a biological process. And I use this diagram, which I took from a review article from Zhishen written in 2003, because I think it captures in a simple way this concept of bottlenecking of populations. And this is a very important biological concept. And in fact, there have been various bottlenecks in the evolution of the malaria, this is falciparum, from its primate ancestors through to modern day parasites. And now we're introducing massive changes in the parasite population, putting stress on the population to reduce the numbers, and only the survivor parasites under these conditions are going to thrive. And of course, one way to test that is to see, can we in fact see evidence for this evolutionary bottleneck in the parasite populations. And this is work from my own lab, in which we've been looking using genetic markers or whole genome sequencing under different conditions of malaria transmission, going from high transmission to low transmission. You see a reduction in the number of parasite genomes, and you see changes in the parasite genomes and their relatedness down to single or small numbers of populations. This data is again published, so I won't go over it in detail, but in fact, you can actually map, this is in Chess, Senegal, with our collaborator Dyann at Czech Antioch University, led by Sarah Volkman and Rachel Daniels. You can see actually by just plotting clonal parasites over time, over years, that in fact in the kind of middle 2000s, after introduction of RDTs and ITNs and IRS at high levels, you saw a bottlenecking and increase in clones, and then things perhaps fell off a little bit, but then reintroduction of these measures, and one can detect these signals with great precision using genetics. Now, in fact, so this technology is available. It allows us to track what's going on in detail, and there are several presentations during the meeting from many groups talking about these approaches. But it's interesting, as I was going through Alan's deck in preparation for this, actually Alan sort of anticipated these results even before they existed in saying, you know, if you had a marker where you could tag parasites, you could barcode them, you could, identify, have a zip code for each and every parasite, you might be able to understand something about transmission and movement of parasites. And indeed, he laid that out in some detail in his thinking about how to approach eradication and with the evolutionary principles in mind. Well, in fact, from the UCSF group, from Brian Greenhouse, in fact, they're exactly applying these kinds of approaches to understand transmission networks in Swaziland. And so I think, again, I want to end now with just probably the final tool that's the most important one for elimination and the eradication. It's not really a new tool, but it's a tool we need to use every day, and that is the educational tool, the sharing of information. And this is a picture of Alan teaching in the Science of Eradication course only a few months before he passed away, where he was sharing his vision as he had every year. This is a course that Pedro, IS Global, and Marcel Tanner from Swiss TPH and my group at Harvard have run now for the past 5 years. And Alan was an active participant in that course, helping us shape what kinds of information the leaders that were going to take the baton, take it to the next step, needed to have. And that is probably among the most important legacies. And I just end where Pedro and Bruno started, with the stamps commemorating the eradication of malaria. This is a humbling slide. These stamps were printed to celebrate malaria elimination in the previous campaign. With that, I'll end. Thank you.